The overall goal of this method is to identify, characterize, and isolate cells from a multicellular spheroid with respect to their cell cycle status and position within the spheroid.
Compared to 2D cell culture, the 3D spheroid model recapitulates the biology of cancer in vivo as it mimics the tumor architecture and its microenvironment.
By pairing spheroids with flow cytometry and imaging techniques, this method can answer key questions in the cancer field, such as how the tumor microenvironment alters drug sensitivity, cell motility, and proliferation.
The main advantage of this method is that it enables real-time visualization of the cell cycle, and it allows for purification of live cells from a specific spheroid region. Demonstrating the procedure will be Danae Sharp, a research assistant from our laboratory.
Begin preparing for 3D spheroid formation by microwaving 1.5% agarose in 100 milliliters of Hanks balanced salt solution, or HBSS, for 3 to 5 minutes. Intermittently swirl the flask to ensure that the agarose is fully dissolved. Then, immediately pipette 100 microliters of the agarose solution into each well of a flat-bottomed 96-well tissue culture plate. Incubate the plate at room temperature for 1 hour to allow the agarose to solidify. Next, remove an 80% confluent cell culture of melanoma cells expressing the FUCCI constructs from the cell culture incubator. Wash the cells with 10 milliliters of HBSS. Add 1.5 milliliters of 0.05% tryptophan.